All right, now to go over the answers. I'm not just going to give you the answers, A, C, B, B. No, I'm going to tell you why. So here, this is a pedigree chart right here. I know we didn't get to practice it, but who cares? They're pretty easy. All right, so number one, the genes for blue eyes are located on chromosome 15 and 19 and are equally as common in females and males. Knowing this, which type of trait is eye color? Well, we know that chromosome number 1 through 22 are the autosomes. And chromosome pair number 23 are the sex chromosomes. So since it's on 15 and 19, that's within 1 through 22. Those are the autosomes. So we know it's an autosomal trait. So now we know it's either going to be A or B. It's not sex-linked because sex-linked is on the 23rd pair. And since it has more than, it's on more than just one chromosome, we know it's autosomal, which means it's on chromosome pair number 1 through 22. And since it's on more than one chromosome, so it's got more than one gene, we also know it's polygenetic. 2 should be very easy. This is just a complete dominance pattern. So what would be the genotype for somebody with blue eyes? Since blue eyes is recessive, the only way you could have blue eyes is you got to be D, little b, little b. Because if you have one big B, you're going to have not blue eyes or brown eyes. Number 3, using the pedigree chart, what is the genotype of the individual labeled 2-1? 2-1 is this girl right here. Remember, circles are girls, squares are the guys. So we need to know what's that person's genotype. What's that girl's genotype? Well, she's not shaded in. Her circle is not shaded in, so we know she does not have blue eyes. So she's got to have at least one big B. Only question is, what's her other B? Is it going to be big or is it going to be little? Well, we can look at her kids. Her kids here are shaded in. So we know 3 of her kids are little b, little b. Well, remember, where do we get these alleles from? We get them from our parents. And we get 1 from each parent. So we can see the guy here is shaded his square, so he's little b, but he can only give 1 little b to his kids. And since 3 of his kids are little b, we know that this girl right here, she has to have the genotype of big B, little b, because she has to have a little b that she gave to her kids to give them blue eyes. So the person labeled 2-1, she's got to be heterozygous. She's got to be big B, little b. 4, using the pedigree chart, what would the probability of the 2 individuals in generation 1? These 2 are generation 1. What's the probability of them having a kid with blue eyes? Well, first we have to figure out the genotype of those 2 individuals that are in generation 1. These 2 people right here. Both of their boxes are not shaded. So we know both of them have at least 1 big B. The question is, what is their other allele? What's their other letter? Well, all you got to do is look at this kid right there. That kid, that boy, his box is shaded in. So we know that boy has the genotype of little b, little b. He had to get both those little b's from both of his parents, 1 from each parent. So, since he had to get a little b from 1 of each parent, we know the other parent has to be, both the parents in generation 1, they have to be big B, little b. And you can do a Punnett square on your own, but it's going to be 25% will be big B, 50% will be big B, little b, and 25% will have the genotype of little b, little b. So their chance of having a kid with blue eyes would be 25%. All right, page 2. Number 5, in this pedigree chart, only females can be carrier. Only females. So there's a difference between males and females. Since there's a difference, we know that these have to be on the sex chromosomes. Since they're on the sex chromosomes, we know it's going to be sex-linked. Now, since they're carriers, the only way you can be a carrier of a trait or a disease and not have it is it's got to be recessive. Because if it was dominant, if you carried the dominant allele, you would not be a carrier. You would have that trait or you would have that disease. But when it's recessive, you can carry that recessive allele for that trait and not have the trait or not have the disease. Number 6. In the pedigree chart, what is the genotype of the male labeled 1? Boom. There he is. He's colorblind. That's a him because it's a square. So we know, first of all, he's XY because this is sex-linked. And we're using B's. So since he's colorblind, he's going to be X little b, Y, nothing. Remember, nothing on the Y. So which one is that? That's going to be E. X little b, Y. That's his genotype. 7. In the pedigree chart, what is the genotype of the female labeled 2? She's right here. It's a female because she's a circle. You can see it's half shaded, half not. So it looks like she's a carrier of the colorblind, but she's not colorblind herself. So first she's a female, so she's XX. She's a carrier, so she's going to have one big N. Excuse me, it's going to be a B, not an N. One big B, one little b. So that would be C. She's X big B, X little b, because she's a carrier of it, but she's not colorblind. And number 8, it says, in the pedigree chart, the male labeled 3, so it's this guy right here. He is colorblind. He's a square, tells you it's a guy. It's shaded in, tells you he's colorblind. Which of the following is true regarding how he acquired / got this trait? Well, you can see his parents right here. His father, the square, was not colorblind, so that's his genotype, X, big N, Y. And you can see his mom was a carrier for it, but she was also not colorblind. So you can see those are her. That's her genotype, X, big N, X, little n. Well, the boy that was colorblind, that's his genotype, X little n, Y. So which one of these described how he got that trait? You can see he got the colorblind allele from his mom, and he just got the kind of a useless Y chromosome from his dad. So which one is this going to be? Receive allele. He did not receive an allele from his father on the Y chromosome, because remember, the Y chromosome does not have an allele for colorblindness. He didn't get a dominant allele from his mom. That's false. He did receive a recessive allele from his mom on the X chromosome. That's true. And he did get no allele from his father on the Y chromosome. That is true. C is the correct answer. All right, in the pedigree chart, a husband and wife are circled in red. That was on the previous page, but they look like this. So it was a carrier female and a colorblind male. So what would be the expected phenotypic ratio of their kids? Draw a Punnett square if necessary, so I drew it for you right here. So we can see that this girl right here, she's going to be X big N, X little n, so she's going to be normal. This girl right here, she's going to be X little n, X little n. She's going to be colorblind. So 50% of the girls are going to be colorblind. This male right here, X big N, Y, he's going to be totally normal male. And this male right here, he's going to be a colorblind male. So the answer would be, it should be 50-50. 50% of males will be colorblind, and 50% of the females will be colorblind on average. 10. Which of the following would correctly describe the term non-disjunction? Remember, junction means to come together. Disjunction means to go apart. Non-disjunction means to stay together again. So that is C. A chromosomal disorder in which chromosomes fail to separate from each other during meiosis. 11. Down syndrome is a disorder caused by an individual being trisomy, having 3 of chromosome number 21. This is caused by, of course, non-disjunction. The chromosomes failed to come apart during meiosis, so the kid ended up having 3 of chromosome number 21 instead of the normal 2. 12. How can you describe the term mutagen? That's not on our test, So boop, boop. I'll cross that one out. 13. Which of the following are true regarding sex-linked traits? Select all that apply. Oh, shoot. They're definitely not more common in females. They're more common in males, because males only have one allele for these traits. And the alleles are usually found on the larger X chromosome, not the smaller Y. And definitely not E, because they're on the sex chromosomes, not on the autosomes. 14, germ mutations, not on the test, cross it out. All right, 15, a mutation which inserts or deletes a base from DNA can be especially damaging. That's called a frameshift, but once again, not on this test. 16, one way scientists can determine where genes are located on chromosomes is to calculate how often they are separated from each other during the process of, crossing over, B. 17 is not on the test, cross it out. Which Mendel's principles suggest that genes for different traits separate without influencing how other traits separate during meiosis, which means it's random. That's not the principle covert, who's our principle. That is the principle of independent assortment, B. 19. Different forms of genes. The different types of genes are called alleles, of course. 20. Which of the following shows a cross between a hybrid and a true breed recessive? Hybrid, we know, means different because it does not have 4 letters in the prefix. True has 4 letters, so we know that means the same. Recessive means it's they're both little. So which one has the first one different and the next one are both little? So that'd be this guy right here. Big B, little b, and a little b, little b. 21. Which of the following shows a cross between a homozygous dominant? Homo has 4 letters. 4 always equals same. And since it's dominant, they're both going to be big. So that's going to be big, big. Hetero does not have 4 letters, so we know that's going to be different. So it's going to be big, big, and then different. Which one's big, big, and different? That would be B. Big R with a big R, little r. Okay, 22. If you cross a hybrid brown-eyed person and a heterozygous brown-eyed person, so we're using B's, what would be the expected genotypic ratio of the offspring? All right, hybrid does not have 4 letters, so we know that means different. That's going to be big B, little b. Hetero prefix has more than 4 letters, so we know that's going to be different again. Big B, little b. So we're going to have big B, big B, big B, little b, big B, little b, and little b, little b. Genotypic. Genotypes are the letters, so it's not brown or blue. So it looks like it's going to be 25% big B, 50% big B, little b, and 25% little b, little b. 23. According to Mendel's principle of segregation, remember segregation means separation. So which one says separate? It's going to be this one. Alleles for the same trait will go into separate gametes. They'll separate. 24. How many different allele combinations can you get from big R, big T, little t? Well, you can get big R, big T, big R, little t, little r, big T, little r, little t. That'd be 4. 25. A rancher bred a homozygous red bull with a true breed white cow producing offspring that have patches of red and white. We should know by now that that's an example of co-dominance, not incomplete dominance. If it was incomplete, you'd have a pink cow, but we don't have pink cows, but we do have red and white cows. If you cross a red flower with a white flower and you got pink flowers, now that would be incomplete dominance. 27, human blood types have 3 different alleles. 3 is more than 2. We usually have 2, but since we have 3 alleles, that's multiple allelic, A. 28, which blood type in humans is dominant? I know type A is dominant, and B is dominant, but since A and B are dominant, hey, it's E. Type A and B are both dominant. Remember, type O is recessive. O is recessive, A and B are both dominant. 29, which of the following would be the genotype for someone with heterozygous? Hetero does not have 4 letters, so that means different. Type B blood. It's not that because we don't have the eyes. We need eyes. It's not that because those are both the same. That's IB. That's not heterozygous. That's homozygous type B. It's not that, because that's type AB blood. That is heterozygous type B. Big IB, little I. 30, a blank mutation. Don't need that. We're going to do that later. Segment of DNA which codes for a specific trait. We call that a gene. 32, the blank is the combination of alleles that an organism has, while the blank is its physical characteristic. That would be A. The genotype is the combination of alleles, while the phenotype is what you can see, the trait. 33, another way to describe an organism's physical trait is, of course, its phenotype. 34, how could you describe big B, little b? Well, obviously those are different. True has 4 letters. That's wrong. Hybrid does not. So that's correct. Pure has 4 letters. That's wrong. Homo has 4 letters. So that's wrong. Hetero has more. So that's going to be right. So it's hybrid or heterozygous describes that. How would you describe big R, big R? Those are both the same. Homo has 4 letters. So we know that's correct. Hetero has more than 4 letters. So that's wrong. True has 4 letters. That means the same, so that's correct. Hybrid has more than 4 letters, so that's wrong. Pure has 4 letters, so we know that's right. Boom, that's a 3-pointer right there. Get that one right, if it's on the test. I don't know. And now the written part. So our regular test will look very similar to this. It'll just be different things, but not too much different. All right, so A, some plants show an incomplete dominant hereditary pattern. Show a cross between a homozygous red and a true breed white. So red is going to be big R, big R. Remember, there's no little letters anymore. This is incomplete dominance. White's going to be WW. So it's going to be RR crossed with a WW. They're all going to be RW. All of them are going to be RW. So, genotypic ratio. How many of them are all RW? That'll be 100%. Or 4 out of 4 will have the genotype of RW. Since it's incomplete dominance and not codominance, all of these things, 100% or 4 out of 4, are going to be pink. A blending between red and white. This one's going to be the opposite, codominant inheritance pattern. True breed purple is going to be big P to be purple. And a homozygous white, that'll be big W, big W. So that's going to be a PP crossed with a WW. So all of them are going to be PW. That's an R, but pretend like that's P. PW, 100%. Genotypic ratio, 100%. We'll have the genotype of PW. And what will these guys look like? All of them are going to look like, since it's codominant, and not incomplete dominant, they're all going to have patches of purple and white. They're going to be purple and white. Okay, what do we got here? The gene for deafness in some cats is sex-linked. Sex-linked. As soon as you see sex-linked, you know we're going to need XX and we're going to need XY. And it's carried on the X chromosome, like usual. A female cat that is heterozygous for deafness is crossed with a male cat who is not deaf. All right, so first, female cat, she's going to be XX, and she's a carrier. So we're using H's. So she's going to be X big H, X little h. The male cat, he's XY, and he's not deaf, so he's going to be big H. So then you just fill it in, X big H, X little h, X big H, Y. This is going to be X big H, totally normal female cat. X big H, X little h, another normal, but she's a carrier. This male cat's going to be X big H, Y. Normal cat, he's not deaf. And this poor male cat, X little h, Y. So the genotypic ratio, I'm not going to write them all, but it's a quarter of each. Phenotypic ratio, 50% of the male cats will be deaf. 50% of the male cats will be normal. And 100% of the female cats will not be deaf. All right, last one. Blood typing. Remember, blood type, we have 3 different alleles. The type A allele is big I A. The type B allele is big I B. And the type O allele is little I. So we're crossing a heterozygous mother. Doesn't matter if it's a mother or female. This is not sex-linked. So she's heterozygous for type A. So she's going to be big I A. And heterozygous, it can't be big I A again. She's got to have a little I. So we'll put her right here. Big I A, little I. And the father is type AB. So his genotype is going to be big I A and big I B. We'll put him right here. Big I A, big I B. Then you just fill them in. It's going to be real hard to do. I don't have much room. Big I A, little I. Big I A, big I B. So this child right here will be big I A, homozygous type A. This kid right here is going to be big I A, little I, heterozygous type A. This kid right here will be big I A, big I B. And this kid right here will be big I B, little I. Genotypic ratios, you just got to write all that. Quarter will be big I A, big I A. Quarter will, 25% will be big I A, little I. 25% will be big I A, big I B. And a quarter or 25% will be big I B, little I. So what's the phenotypic ratio? These two up here, they're both type A. One's heterozygous, and the other one's homozygous, but 50% will be type A. And this one right here is type AB. So 25% will be type AB. And the last 25% will be type B. And the last one, construct a chromosome gene map given the following data. All right, remember, we always start with the biggest number. M and Q are separated from each other during crossing over 11% of the time. So what you do is you're just going to pick anywhere. Pick right there. And you will label that M. Now you know that Q is going to be 11 lines away. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Boom. There's Q. Now you can see here that A is going to be 8 lines away from Q. So you go back the other way. 1, 2, 3, 4, 5, 6, 7, 8. Boop. There's A. And the only one we have left is Y. We can see Y is 5.5 away from M. So that'd be 1, 2, 3, 4, 5.5. So that would be like right there. And that would be Y. All right. That is it. Hopefully this helped you out. Remember, if you're still stressed out, now if you don't care about your grade, you never get stressed out. But if you do care about your grade and you really want to do well on this exam, feel free to come in Friday at lunch, and I will go over anything you need to. All right, thank you for listening, and I will see you guys next week, or Friday at lunch if you come in.